Dr. Mohammad Heidaran, who's part of the Office of Cellular, Tissue, and Gene Therapies from CBER, FDA. He's currently a CMC reviewer of CBER at the Office of Cellular, Tissue, and Gene Therapies. His in-depth experience in evaluating investigational new drugs, INDs, and biological trade applications, BLAs, come from both product review and good current manufacturing practices. Anyway, Mo, it's your turn on the podium.
On behalf of our office, I would like to thank the organizers for their invitations. My talk today will be divided into three parts. I will give you a brief introduction to our office. I will give you some updates of recent development. And finally, I wanted to emphasize the importance of establishing manufacturing consistency as part of the drug development cycle. OCTG regulated products are shown in this slide. Are you probably familiar with the cellular and gene therapy products? But we also oversee cancer vaccine and immunotherapy consisting of bacterial mammalian cells, gene modified or not, peptides, and protein-based products. We also oversee tissue-based product, xenotransplantation, and in regard to devices and combination products, some devices with cellular component, and also devices manufacturing or delivery of cells. This is the list of licensed products from our office. You're familiar with Provenge and Carticel, but Lviv is also one. Theracide is actually a BCG vaccine for treatment of urinary bladder cancer. Gentuit, and we have so far licensed 5 cord blood manufacturers. Recent update, quickly, since I don't have a lot of time. As of August 8, 2013, wound care products with live cells are essentially transferred to our office from CDRH. With respect to new guidances, we have published 4 new guidances, 2 on cord blood and 1 on consideration of the design of early phase clinical trial and 1 on preclinical assessment for investigational drugs. PDUFA 5, which is applicable between 2012, October 1st of 2012 to September 30 of 2017. The major difference in PDUFA 5 is the timeline for BLA, which is now 12 months, with the first 2 months used to conduct filing review. Priority review, 8 months, with the first 2 months used to conduct filing review. And there is some minor changes with respect to time left of different formal meeting. At this time, I wanted to emphasize a recent publication from FDA and CBER in regard to process validation and give you an update on that as well. The new process validation, which was published in 2011, conveys FDA's current thinking on process validation and is somewhat different from publication in 1987. There is no reference to retrospective process validation, and process validation is essentially defined very clearly in this document. I encourage you to take a look at it. Essentially, from that perspective, the document states that the manufacturers should understand the source of variation, detect the presence and degree of variation, understand the impact of variation on the process, and control the variation in a manner that would result in a high quality product. It also introduces a product life cycle approach to process validation and talk about different stages from process design, process qualification, and continued process verification. I encourage you to take a look at this document. In regard to sterility, we have amended the sterility regulation which eliminate the specific sterility methods, culture media formula, and culture media test requirement. So we don't have a specific test for sterility. If you use non-compendial method, full validation and subsequent verifications are required. If you use the traditional method, compendial, only verification is required. In regard to the accelerated availability of drug, I encourage you to take a look at the following document. I'm not going to talk about fast track, accelerated approval, or priority review, but a little bit about our recent announcement on breakthrough therapy, which is shown in the following document. For breakthrough therapy designation, again, is intended to expedite the development and review of drugs for serious life-threatening. Preliminary clinical evidence is required. With breakthrough therapy provides is regular, more frequent sponsor meeting. A smaller patient population is applicable, and you have more interactive interaction with FDA. It is not accelerated approval or priority review, but those programs also may be applicable. A word about cost recovery, which is something that we also see, intended to allow IND sponsors to continue providing clinical trial by covering the direct cost of the manufacturing. Two keywords: that the clinical trial must demonstrate clinical benefit to the patient, and that the direct cost should be calculated, and the document for the direct cost has to be accompanied by a statement by an independent certified public accountant that that document and calculation has been reviewed and approved in your application. The cost recovery essentially allows you to charge for the direct cost of the manufacturing. Again, clinical benefit has to be demonstrated, direct cost has to be listed, and the certified public accountant information has to be provided. And you have to demonstrate that you cannot continue doing the clinical trial if the cost has not been recovered. Of note, FDA has no authority over the third-party payers, private or public in this case. A quick note about establishing manufacturing consistency, which is very important to me as in this meeting. Given that every product has a set and predefined threshold for product quality, it is reasonable to conclude that the efficacy of a product can be more readily established when the product can be manufactured consistently with high quality, as opposed with a product that cannot be manufactured consistently. The source of inconsistency in the product are many. Despite the progress we have made in the manufacturing, these sources of variability include variability of cell source, reagent material, the complexity involved in cell culturing and cell manufacturing, and the signaling involved in these processes that are highly complex and convoluted, and incomplete knowledge of the product attributes and process parameters as we go forward. Minimizing these variabilities requires not only our knowledge of the process validation, but also special attention to knowing your product and applying good manufacturing practices as early as possible. A little bit about knowing your product. Knowing your product means that for any given product, you must know the critical quality attributes of the product. the critical process parameters for manufacturing that product, and that you have a good knowledge of performing risk assessment in order to link critical quality attributes to the process parameters. So what is the critical quality attribute? A brief introduction is defined in the Q6B, ICH, is biological and molecular characteristic found to be useful in measuring the safety and efficacy of the product. Note the emphasis on efficacy. While manufacturers and sponsors start with a spectrum of the quality attributes for their products, we expect that only a few of these attributes to be critical to the quality of the product. Potency assay is an example of measuring one of the critical quality attributes of the product. And it could be any in vitro or in vivo assays that measures the relevant biological activity of the product. Regulation is very flexible in regard to the potency assay. We don't have a pre-prescribed expectation from the regulatory perspective of what the potency should be and what it should measure. For this reason, we encourage you to, early on in clinical trial, to think about selecting multiple assays for potency assays. And by phase 3 of the trial, that the potency assay should be validated. A little bit about critical process parameters, which are defined again in the ICH document as independent process step or parameters most likely to affect the quality attributes of a product. So the critical process parameters have to be linked to the quality attributes of the product. And this is highly critical. Since we have limited knowledge in regard to cell therapy products in respect to the CQA and CPP, risk assessment becomes an important part of the product development cycle. Risk assessment is essential for us because it provides you tools to identify and rank parameters with potential impact to product quality. We need to know that when you submit your application. So other than knowing your product, the second step is being aware of the current good manufacturing practices. For knowledge in regard to the phase 1 requirement for current good manufacturing practices, you may refer to the following documents. GMPs are a set of good manufacturing practices defined here that results in a high quality product. It prevents cross-contamination and also prevent product contamination with foreign material. These foreign material could be particulate, visible or non-visible, or they could be adventitious agents that are highly undesirable. But current good manufacturing practices in cell therapy products are going to result in manufacturing consistency of high quality product, in our opinion. General CGMP requirements are shown here. They expand from personnel to record keeping, but the quality control is probably the most important aspect of CGMPs. Quality control units in your manufacturing facilities, their responsibilities are outlined here. The responsibilities are to review and release component, review and approve production procedure, release or reject each batch upon cumulative review, and investigate errors and initiate corrective actions. The responsibilities are performed independent of production and has to be performed by trained individual. CGMP, full CGMPs, of course, is verified at the time of pre-licensure inspection. CGMP is verified at that time according to the PHS Act, Section 351, and FTC Act. And you have to be in compliance with Federal Regulation 21 CFR 210-211, 600, and 1271, human cell and tissue cellular product. This slide highlights some of this, what we use, which is a system approach for the inspection processes according to our compliance program. We looked at 7 key systems: quality system, production, facility equipment, material, packaging, laboratory, donor eligibility, which is highly critical. And in each key system, we look at 3 critical elements: standard operating procedure, training, and documentation. This slide, which is essentially one to the last slide, is designed to give you a heads up on some of the inspectional items that we have identified in our already licensed manufacturers. These include inadequate quality system. That's a common one that we see. Quality control unit lacks responsibility and authority, and procedure and documentation. Quality unit is very critical. And incomplete process validation, and that's the reason why I discussed and gave you a reference to our process validation documentation to take a look at, which also include reference to aseptic process validation. Incomplete batch record is also cited, and that seems to be a common problem as well. Also, there's a lack of cross-contamination control, such as inadequate space when operations are performed. And there's inadequate segregation of the quarantine material. And of course, there is a learning curve that we see in manufacturing companies in regard to label and label reconciliation. So that's something I can talk to you about later on. So becoming CGMP compliant is a process, and it is never too early to plan for that process. It takes time, and we recommend that you start as early as possible in your drug development cycle. The last slide, I want to emphasize that I appreciate the desire for getting these products to market faster, but really the whole process can be accelerated by applying good manufacturing process, by knowing your product more fully, and to understand the risk associated with those uncertainty. So please, we encourage you to take a look at the documentation in this slide deck that I can provide you later on. Please understand the critical quality attribute of your product. Those are key attributes that regulate and are linked to the quality of the product. And also, it is important to have a good knowledge of the critical process parameters. Those 2 things are essentially very important, not only for developing your product and getting approval of your product, but also to doing post-approval changes, because inevitably you will change your product. And when you want to change your product, if you have control over these parameters, the change and the comparability and the data that is needed is going to be extremely streamlined. So it is to your benefit. And also conduct full risk assessment as shown in this slide. Because our knowledge is limited, risk assessment becomes an integral part of any application, because we have an incomplete understanding of some of the product quality issues. Optimize and apply CGMP's current good manufacturings for your product early on as possible. If you would like more information, of course, you can reach us at any time, and you can also reach us through formal meeting as shown in this slide. We are very open to have discussion around questions you have through formal meeting. And this is some of the relevant guidances. And if you want to contact me to provide you with a copy of this slide deck, please feel free to do so. Thank you.